Nucleus. General features. Nucleus is an essential organelle present in all viable cells. It is absent in mature mammalian erythrocytes, RBCs, and blood platelets. These are not considered true cells. Site. Nucleus may be central or eccentric or peripheral in position. Number. Usually, each cell contains one nucleus, mononucleated, but some cells may contain two nuclei, binucleated, as liver cells. Others may contain more than two nuclei, multinucleated, as osteoclasts in bone. The nucleus varies in size. It may be large or small. Also, its shape is different. It may be flat, round, oval, or kidney-shaped. Sometimes the nucleus may be lobulated, as in neutrophils. Types. The nucleus may be either vesicular or called open-face nucleus, which appears pale with few chromatin and excess nuclear sap, as in liver and nerve cells, or condensed nucleus, which appears dark with clumped chromatin and less nuclear sap, as in lymphocytes. Structure. Nucleus consists of nuclear membrane, chromatin network, nucleolus, and nuclear sap. Nuclear membrane, by light microscope, appears basophilic due to attached chromatin granules on its inner layer. Chromatin is formed of desoxyribonucleic acid, so it accepts the basophilic color of hematoxylin, as it is acidic in nature. By electron microscope, nuclear membrane is a double-walled unit membrane with many pores. Its inner layer is smooth, lined by chromatin, while its outer layer is rough, covered by ribosomes, and is continuous with the endoplasmic reticulum. Functions of the nuclear membrane. It maintains the shape of the nucleus. It is permeable to macromolecules less than 10 nanometers through the pores by passive transport, as molecules of messenger ribonucleic acid and formed protein. Large molecules more than 10 nanometer needs energy-dependent transport mechanism and needs the activity of certain enzymes. Nuclear pore complex. Pores are formed at sites with the inner and outer membranes of the nuclear envelope are joined together. Pores has an effective diameter of about 10 nanometer and are closed by nuclear pore complex, NPC, which allows the exchange of substances between the cytoplasm and the nucleus. Structure. It is a tubular structure and its wall contains 8 subunits and has 2 rings, one on each of its ends. Outer ring is attached to outer membrane and inner ring is attached to the inner membrane of the nuclear envelope. 8 radial spokes extends inwards from the subunits towards a central granule or plug. The radial spokes and central granule are responsible for false appearance of pore diaphragm in micrographs and form the principal barrier to movement through the pores in the two-way traffic
between the nucleus and cytoplasm.
Chromatin consists mainly of nucleoprotein formed of desoxyribonucleic acid, DNA, combined with other proteins as histones. By light microscope, chromatin material appears as coarse or fine, dark basophilic granules and filaments. By electron microscope, two types of chromatin can be distinguished according to the activity of the cell and the arrangement of chromatin material. Euchromatin, or extended or active chromatin, which appears pale and is formed of thin, uncoiled chromatin threads carrying the active gene. It is predominant in metabolically active cells as in nerve cells. Heterochromatin is also called condensed or inactive chromatin. It appears dark and is formed of coiled, clumped chromatin threads carrying the inactive genes. It is predominant in metabolically inactive cells like fibrocytes. Heterochromatin is given name according to the site or location in the nucleus. There are three types of heterochromatin. Peripheral chromatin attached to the inner wall of the nuclear membrane. Nucleolus associated chromatin attached around the nucleolus. Island chromatin scattered inside the nucleus. Functions of chromatin. Chromatin carries the genetic informations. It controls the protein synthesis in the cytoplasm. Nucleolus. Nucleoli are found only in the interphase and are prominent in cells actively synthesizing proteins. Nucleoli may be single or double. Nucleolus disperses or disappears during cell divisions and reappears once more in the telophase stage of mitosis. By light microscope, nucleolus is a rounded, darkly basophilic structure. Its basophilia is partially due to attached nucleolus-associated chromatin and partially due to its contents of ribonucleic acid and protein. By electron microscope, nucleolus has a sponge-like appearance, showing dark material permeated by light spaces filled with sap. The dark material consists of granular part or bars granulosa, formed of granules of ribonucleic acid. Fibrillar part or bars fibrosa, formed of filaments of both ribonucleic acid and desoxyribonucleic acid. Function of nucleolus. Nucleolus is mainly concerned with the formation of ribosomal ribonucleic acid. Nuclear sap, karyolymph, appears as a colorless colloid solution in which chromatin is embedded. Nuclear sap contains nucleoproteins, enzymes, sugars, and minerals. According to the amount of nuclear sap in relation to chromatin content, nuclei are distinguished as two types: open phase or vesicular nucleus, where there is excess amount of sap in relation to chromatin content. The nucleus appears pale as in liver cells. Condensed nucleus, where there is less amount of sap in relation to chromatin content. The nucleus appears dark as in lymphocytes. Function of nuclear sap. It acts as a medium for the movement of ribonucleic acid towards the nuclear pores. Functions of the nucleus. The nucleus is important for vitality and division of the cell. It is the site of storage of genetic informations. Also, it is the site for formation of three types of ribonucleic acids: ribosomal ribonucleic acid, transfer ribonucleic acids, and messenger ribonucleic acid.
Nuclear signs of cell death. The nucleus becomes smaller in size and deeply stained by kenosis, then breaks down into smaller fragments, karyolysis, and finally dissolves and disappears, karyolysis.
Nucleic acids. Nucleic acids are important and essential for vitality of the cells. They are of two types: desoxyribonucleic acid DNA and ribonucleic acid RNA. Desoxyribonucleic acid DNA site. It is found mainly in the nucleus chromatin, but also very small amounts are found in the mitochondria. Structure. DNA molecules are made of two twisted strands or chains forming a double helix. Each strand is made of alternating pentose sugar, desoxyribose, and phosphate. The two strands are linked transversely by four nitrogenous bases, extending laterally from each sugar group. The nitrogenous bases are adenine, thymine, guanine, and cytosine. Adenine is always linked to thymine, while guanine is always linked to cytosine by a hydrogen bond. This linkage is very important to ensure the formation of an exact copy of DNA molecule during its replication or duplication in the interphase before cell division. Functions of DNA molecules. Storage and carriage of genetic informations. Replication or duplication of DNA molecules during the interphase before cell division, thus keeping the amount of DNA molecules constant in daughter cells. Formation of three types of ribonucleic acids: ribosomal ribonucleic acid, transfer ribonucleic acid, and messenger ribonucleic acid by the process of transcription. By this, they transferred the genetic informations. Ribonucleic acid RNA site. It is found mainly in the cytoplasm. It is formed in the nucleolus from DNA molecules by the process of transcription. Structure. RNA molecules are basically similar to DNA molecules, except RNA molecules are made of a single strand or helix. Some regions, however, may have double helix. Each strand is made of alternating ribose sugar, not desoxyribose, and phosphate. The four nitrogenous bases are adenine, uracil instead of thymine, guanine, and cytosine, extending from the sugar group. Adenine is always joined to uracil, while guanine is always joined to cytosine by a hydrogen bond. There are three types of ribonucleic acid, RNA, according to function: messenger ribonucleic acid, messenger RNA, formed from DNA molecules in the nucleus, carries from DNA molecules the genetic informations or information codes needed for protein synthesis to ribosomes or factories for protein synthesis in the cytoplasm. Transfer ribonucleic acid, transfer RNA, also formed from a special type of DNA molecules in the nucleus and is transported to the cytoplasm, where it transfers the specific amino acids needed for protein synthesis on their proper places to ribosomes or protein factories. Ribosomal ribonucleic acid, ribosomal RNA, also formed from DNA molecules in the nucleolus. The newly formed ribosomal RNA molecules, once they reach the cytoplasm, they are called ribosomes and are the sites for the protein synthesis or the factories for protein synthesis. To summarize the steps for protein synthesis, ribosomal RNA collects messages from messenger RNA and specific amino acids from transfer RNA, then manufacture the needed proteins.